So now we've got the pleasure of having Dr. Aylwyn Scally from the University of Cambridge, who's researched on evolutionary genomic and demographic processes responsible for present-day genetic diversity of species and individuals, and particularly mainly primate and human evolution. Yes. And his talk is entitled Evolutionary Genomics of Survival and Extinction in the Great Apes.
Okay. Let's get this up. Great. So I never had the pleasure of meeting Andrew Sheppy, but I very much enjoyed hearing all about his legacy and the work he's inspired. And I hope, so what I'm going to talk about is I'm delighted to have the opportunity to come and speak. I'm going to talk about something which is a little bit different. I'm not going to talk about domesticated animals. But I think that a lot of the ideas and approaches that I will discuss may be of interest and perhaps relevant to some of the things that people are thinking about. So, so I'm going to begin by way of this, as a sort of prelude, back in Africa in 1902. So here is the continent as it was then, parceled up into regions by the big European colonial powers, regions that they were then going to, or that they were in the process of extorting for resources and wealth. And one region here in particular, the Germans had recently, or 10 years prior to that, or 20, had agreed with the British the boundaries of the region that they were going to particularly exploit. And as part of that, then they wanted to kind of discover what it was that they had claimed. And an example of one of these activities was led by this chap, Robert von Virunga, guy in the white there with his troop of soldiers, Askari, behind him, and they set off to explore this northwestern corner of the German East African claim. And so they set off from this town here at the top of Usumbura, at the top of Lake Tanzania, and headed north. And first thing they did was they met up with what was then called the Sultan of Rwanda, an individual, presumably a tribal leader there, and I imagine sort of implicitly threatened him in some way. And before moving on north, deciding to explore the rest of this landscape, and they reached this kind of region up here, which was the Virunga region in Central Africa. As you can see, it's this rugged, highly forested landscape dominated by this range of active volcanoes. And they decided to go and climb one of these. This is at the sort of the extension of this territory that they were in. So it's a difficult region to navigate even today, and they stopped and made camp on a ridge up one of these mountains, and noticed while they were there that across on the nearby side of the mountain was a group of animals, large monkeys, as Virunga later called them, which he was surprised because he didn't expect to find any large apes or monkeys in this region, and so did what any intrepid explorer would do at this time, and shot them. And so after a few hours' work, they managed to drag one of these, the bodies back up to their camp, and took selfies. And you can see, so this was so here it's not a great picture, but this animal here, they then eventually somehow managed to get shipped back to Berlin for analysis, and it was decided that this was a new species of gorilla, not hitherto known, and named in his honor gorilla Berengay. And so, although it's the Linnean system has many great features, but it's unfortunate that in this and in many other cases it enshrines the name of the first European to kill one of the animals concerned. But, I mean, it's ridiculous in some ways to call this the discovery, of course, because these creatures would have been living in proximity to humans for hundreds of thousands of years, but that's how our system currently works. So I mentioned that he didn't expect to find any apes or gorillas in this region, and it's kind of surprising, really, that as late as the beginning of the 20th century, an entire species of great ape was completely unknown to Europeans at any rate. And it's interesting to know to reflect on what was known about apes back at that time and what we've learned since. So the first person to think about the great apes, or in fact primates, in an evolutionary way, as in many other cases, was Darwin. In 1868 he did this little sketch in his notebook where he decided to try and sketch out what he could make of the tree, the evolutionary tree of the primates. It's a little bit difficult to read, but he has up here humans, or man as he says, on their own long separate branch here, and here we've got gorillas and chimpanzees. It's not clear as to how to distinguish those. Orangutans nearby, so he correctly placed the great, what we call the great apes, together, and the other apes, the gibbons, hylobates, in a clade. But this extra grouping, apart from this fact, the fact that humans he has on an extra long branch, the rest of this tree is pretty much as we still know it today. It's remarkable how accurate this was, given that he was only basing it on morphology and not even, and accounts of morphology, in fact. So quite insightful. And in fact, a tree of this nature with humans on their own long extra branch was actually considered to be, was held to be the correct evolutionary tree right up until the 1960s, actually. And it was only really with the advent of molecular methods, and in particular gene sequencing, gene and protein sequencing, that we were able to establish the correct relationship between these species. So here's the kind of data that I'm talking about, and this is closer effectively to what I do, which has at various times involved looking at this kind of thing. This is just a little fragment of genome sequences from 6 primates. So Homo sapiens at the top, and then you've got chimpanzees, next row is gorillas, orangutans, macaques, and marmosets. And you can see just a little bit, and the little boxes indicate places where the sequences differ from human. And so intuitively, with our concept of the tree, the further away you go, the more evolutionarily distant you go from human, the more genetic differences you are likely to find. And we can essentially count up the number of differences we see across very large stretches of the genome, and we can work out what the genetic divergence is. That's simply the number of differences you see, the percentage of different sequences that are different between these species, or genera in this case. So here we got humans and chimpanzees only differ by about 1.3%, 1.4% on average across the whole genome, or in total, should we say. And a little bit further, a little bit more difference between human and gorilla, and between human and orangutan, and you can measure all the pairwise differences, and you can then, based on these, reconstruct the correct tree, which is that humans are in fact most closely related to chimpanzees, then to gorillas, being an outgroup of those two, and then orangutans further back diverging like this. So that gives us the topology of our relationship with our nearest evolutionary cousins. So that resolves that question. We'd also like to know actually, when and how did these divergences occur? And so I'll go off on a little digression and talk about how we actually put dates on these, because that sort of completes the picture. It's not sufficient to describe their evolution merely to talk about the relationship. You'd also like to know when this happened. And to do that, we have to think a little bit more about how these mutations, these differences arise. Essentially, genetic differences between humans and chimpanzees will correspond to mutations that have occurred on the genetic lineage since the common ancestor. And we can turn that around and say, if we know what the mutation rate, the rate at which mutations accumulate, then we can turn that around and work out what the time was. How long have these mutations been accumulating for? That's a measure of the speciation time between them. And so one way of estimating this mutation rate is actually to say, well, suppose we know the speciation time in a particular case, then we can calibrate our mutation rate based on that. So this starts to sound a little bit circular, but nevertheless, this is what most mutation rates have done, or how most mutation rates are estimated across the animal kingdom. And the way you do it is you take a couple of fossils. Oopsie. The red one here that you putatively assign as ancestral to the speciation time, meaning that it looks about as far, it looks roughly as different from both of the two modern species. So there's no reason to think that it's closer to one or the other. And the blue fossil which you call a divergent, a derived fossil, meaning it's a little bit more like, say, in this case, humans. And since you can often date fossils pretty well, radiometrically or stratigraphically, that gives you, that sort of brackets the speciation time. So then you take your speciation time as the sort of maybe the mean difference between those two, and your mutation rate is the genetic divergence divided by twice that speciation time, because you've got mutations could accumulate on both branches. And so there's your estimate. People have done this for human chimpanzees and estimated a number of 10 to the minus 9 mutations per base pair per year, which is a very, very small number. So mutation rate is very low. They accumulate very slowly. So that we can then turn around, we can use this. Now it seems, as I said, completely circular to then turn around and use this to date the human chimpanzee speciation time. We're just putting back in the numbers we've calculated. But you can extend it and use the same number for humans and gorillas and orangutans and elsewhere in the primate tree. There's a couple of problems with this approach. One is that as a sort of population genetics technicality, it's not the case actually that the speciation time is when your genetic lineages will diverge. They will actually diverge further back in time than the speciation date. So the genetic lineages, which I've drawn by this black line here, actually will find a common ancestor, or coalesce, as we say, further back by some amount, which we call the ancestral coalescent time. So you've got this additional time, which you've no idea how long that was when your mutations were accumulating. It's longer than the speciation time. You have to sort of make that a correction for that somehow, and that involves basically sort of guessing it. And then the other problem is that your fossils might not have been correctly placed. That what you think is an ancestral fossil here might actually be really on a separate, much older branch. I mean, this branch would also, its common ancestor up here would also look the same distance from these two, and therefore you'd place it as an ancestral fossil. But you're massively underestimating the speciation time if you call it here, when in fact it was all the way back up here. This can happen too. So there are problems with this approach. So recently we've been able to take other approaches to estimating this mutation rate, which is looking in modern day humans. You could also do it in other species, comparing the two parents and the offspring and actually seeing how many times do we see a new mutation. This is a position in the genome, in the child's genome, where, for example, here, neither parent carries this C. That's a new mutation that's occurred in that generation. And similarly here, another one. And you can count up all of those. And there's a whole bunch of data processing issues that are problematic because these are actually very rare. But nevertheless, we can do that too. And interestingly, doing this, we find a much lower mutation rate than we had estimated previously from the phylogenetic rate. So there's some kind of question about which is the correct value to use, and has it changed over time? Probably it has. So there's some ambiguity there. And that corresponds to an ambiguity in our timing. But it's worth bearing in mind, actually, that there's also ambiguity in the whole process of speciation and what that means. So here's a nice clean species tree that I've drawn here, the ancestral species, and separating out into two modern-day species. But that's probably rare in nature, actually. There's probably plenty of times when there was a gradual separation over an extended period of time, which could be many millions of years, this period here. When do you say speciation started, and when did it occur? Is it the middle, or do you say it only happened here or something? And it's not uncommon to find much later episodes of gene flow between apparently diverged species too. So the whole concept is inherently a fuzzy one. Okay. So anyway, we end up with values around about, going through this and making appropriate assumptions, we end up with numbers around about this 6 million years ago for human chimpanzees, 10 million years back to our ancestor with gorillas, and 18 with orangutans. And back then, at this point in time, 18 million years ago, was actually a sort of an era of abundance for the apes in general. Planet of the apes, people have called it. And apes, you find ape fossils distributed all over Africa, all over Europe and Eurasia, in places that none of them live now. And this is due to the different climate and landscape that existed back then. And since then, it's really been a story of decline and fragmentation and extinction in most cases. And now, although to humans, because we get misled with it, we're great apes, we're all over the planet, so you could say we're a success story. But every other non-human great ape is living in a small, isolated, fragmented environment in equatorial forests. and most species in our own genus and in other great ape genera have gone extinct. So to zoom in a little bit on the great apes and break them down into their constituent species and subspecies. Here's a picture of where they currently are and with their relationship up above. So in fact there's two gorilla species. There's a western species and an eastern species, the eastern one being the gorilla Berengay that was discovered by our German friend. And then you've got chimpanzees, 3 or 4 chimpanzee species, and subspecies, and then bonobos, another species of the pan genus. Orangutans over here in Southeast Asia, in Sumatra and Borneo, and very recently, actually last year, there was a new orangutan species was described, not discovered in the sense that it was the population was known about, but the determination was made that this should be called a new species. Talk about that in a minute. But anyway, it looks from this that humans are somewhat unusual in that there's only one of us, and we're everywhere. But of course, you don't have to go too far back in history before there are other hominins as well, like Neanderthals and Homo erectus all over the globe, and a little bit further back, and you find that all our ancestors were here similarly in some sort of equatorial region, the other side of the Great Rift. Now, one thing that has dramatically changed this entire field in the last decade or so has been the advent of next generation sequencing, as it used to be called. It's now just called sequencing, genome sequencing, and that has allowed us to collect whole genome sequence data for hundreds of individuals. Well, in the case of humans, tens of thousands or hundreds of thousands of individuals, but for other species, we're only at sort of dozens or hundreds, but much, much more cheaply than it was possible before. Many orders of magnitude more cheaply, and that's enabled us to do population-level studies of some of these isolated and endangered populations. All of these populations are endangered or critically endangered. So, for example, in chimpanzees, people have sequenced samples from all over Africa, and they've been able to, based on where the samples are known to have come from, they're able to place samples of unknown origin into this. So here, that's a potential benefit for conservation genetics, tangible benefit, I should say, from doing this. Orangutans, people have similarly done a study that was published last year and compared all of these species, these populations, I should say, and as I mentioned, by identifying this new species of orangutan, which is this isolated population in southernmost population in Sumatra. Although I should say this identification really is not based on genetic data. It's really a morphological and ecological definition of the species. Genetically, they are not all that different from their northern neighbors. Population I want to focus on is the mountain gorillas, which are extremely, until the classification of this species, were the smallest and most endangered great ape. So they live in this region that I mentioned before, the Virunga region, and a little bit to the north in windy, similar kind of landscape. And there's only a few hundred of them left, about 800 in total, half of which are in Virunga. And so the study that I was involved in, we collected sequences from about 13 mountain gorillas, and then a whole bunch of other ones from nearby, from the eastern species, and other gorillas from the western species, and looked at their genomics. So there's all kinds of really important and useful analyses you can do once you've got whole genome sequence data. And one is just to get a sense of what kind of real population structure there is, because often it's difficult to tell just phenotypically. We know that mountain gorillas are under threat, and there's their habitat. I won't go through everything in this plot, but pretty much everything on this map, which is where they live, is a threat to them. The blue is a civil war that's going on with rebel-held regions. The green is not a threat. That's natural. Those are natural parks. The brown and yellow are mining operations, and there's also logging in these kind of orange areas. So their habitat is under severe threat. This is now an extremely heavily populated part of Africa. And an indication of that is just shown here. These two Landsat images, one from 1978 and one from 2005, and the green represents sort of the natural vegetation. It's color-coded to represent the natural vegetation that lives there, not farmland, in other words. And you can see that essentially it's shrunk right back down. The only thing that's preserved it is the fact that it's a national park and that it's patrolled by rangers. And were they not there, this habitat would be gone, and with it, the mountain gorillas. These kind of incidents still occur. In sort of continuing Virunga's legacy, these gorillas are not infrequently subjected to sort of mass killing on the grounds that their existence essentially supports the national park and therefore prevents local people from being able to use the park's resources. So they're seen as a threat for that way. But probably the biggest threat to gorillas is actually not humans directly, although indirectly we may be involved, but it's the Ebola virus, which only a few, I think a decade ago, wiped out 20,000 western lowland gorillas. Were it to strike in the mountain gorilla region, they'd all be gone within a few months. So this is a natural threat that they face. So you can look at this is one quite interesting way of looking at population structure within animals, like, within any population actually. So what this plot shows is you're looking at chromosome matching between one chromosome and another between two individuals. So all the individuals listed here, and they're colored by species, and so we take one of the individuals here, and you go along and you see going down to here, that's how much the color represents how much of its genome is shared essentially identically by descent with that other individual. And so so this yellow block here corresponds to all the individuals within one particular species. That's or subspecies, the eastern lowland species. These are the mountain gorillas here. They also share even more with each other, you can see, because we're sort of redder colors than yellow here. And the blue, there's a sort of slightly lighter blue here. That's all of the western lowland gorillas. They share more with each other than with the other species, but nevertheless, they still share a lot less. There's more. In other words, the way of saying that is that they're more genetically different from each other. So you can see some substructure in here, and actually this represents sort of actual family, close relationships between some of these individuals. So as well as looking at sharing of chromosomes between individuals, you can look at how much matching there is within the two chromosomes within each individual. And that's basically a measure of inbreeding in that population. And what this plot shows is for every single gorilla in the study, all these western lowlands, eastern lowlands, and mountain gorillas, it's a stretch of the chromosome, and the thick regions are places where the two chromosomes are identical. And so you can see that there's much more of this identity, whole stretches of homozygosity, long stretches, many, many megabases long, in the mountain gorillas, much more than in the eastern lowlands, and certainly much more than in this more outbred group. And in fact, you can go across the whole genome and work out how much in total there is. And it turns out that in mountain gorillas, almost a third of the genome is homozygous in this way. That's an enormous proportion of the genome. When you compare it, for example, in humans, the smallest human populations, the most inbred human populations are populations like the Papuans or Carrotiana in South America, only a few percent, where mountain gorillas are up sort of, 20% here in really long tracks, and over 30% if you consider all the regions, including sort of smaller tracks like this. And eastern lowlands similarly. So this is a legacy of the fact that they're clearly living in a very small population. And what you might ask is the impact of this on their biology and on their genetics. Well, one striking result of this, it's twofold. It's well known that in an inbred population you're going to accumulate, selection gets weaker, and you're more likely to accumulate deleterious alleles, that is alleles that are selectively bad for you, that they're associated with propensity to disease, or they're just not so good. They break things in your genome. And that does happen. So compared to the more outbred population, the western lowlands, this plot shows if you just look at the yellow bars, in each case a bar extending to the right means here that the eastern lowlands have more what are called missense, that is just they change protein changing mutations, than the western lowlands. And protein changing is generally a bad idea. If you just randomly change a protein in your body, the chances are it's not going to be beneficial. So overall, these things are likely to be, missense mutations are likely to be deleterious. And the same is true of the mountain gorillas. But the other thing is, if you look at the really severe mutations, that is things not just that change a protein, but actually will cause it to break totally, like will stop the protein, make that protein not function at all. These are much more likely to be severely deleterious, maybe even lethal, if present in 2 copies. And in those cases, both the mountain gorillas and the eastern gorillas have fewer of them than the outbred population. So what we're saying is here, this is a very inbred population, and it essentially has purged the really severe deleterious mutations from it, due to the fact that they can't hide in a sort of a heterozygous state. You can't, in a large population, this really deleterious mutation can hide in a single copy in individuals and just kind of stay in the population. It's not very likely to find itself in 2. But in a very small population, pretty soon it meets another copy and then gets lost. And so these deleterious, severely deleterious mutations basically get purged as a result. So this was kind of taken as good news when we published this paper, and then the sort of press reports varied, but people saying inbreeding makes mountain gorillas genetically healthy was an example of one story. Various variations on that somehow this was a kind of a good thing for them. And the other versions of this are obvious puns on gorillas in the mist, about how the... you'd think they could have called something a bit better than that. But anyway, and not really. That would be, I mean, it's true to the extent that that is a sort of a silver lining, you might say. That you've lost some of the severely deleterious things, but your overall burden of deleterious mutation has definitely increased. Your genetic diversity is reduced, the ability to adapt to new changes, climate change, for example, or virus infection is reduced. So, for example, here, this is the gorilla immune system compared to humans. So the immune system benefits by being very diverse, and in humans, this is just a graph of how genetically diverse the genomes are in this key region where the immune system is encoded. Humans are represented by these orange and red curves, and you can see there's big spikes here where the immune system proteins are coded. There's lots of diversity. Gorillas, the blue, green, and brown, are much, much lower. Really, they're no more diverse here than they are elsewhere in the genome. So that suggests that they've lost a lot or have never gained the genetic diversity in that region. Their immune systems are essentially quite similar. Whether or not, how bad that is, we don't really know, but nevertheless, it suggests it might be a problem. But there is one aspect in which the news might not be as bad as we think, in that gorillas, this is kind of, I'm wrapping up really, gorillas, well, what you can do is you can make an analysis of the ancestral effective population size. This is the history of effective population size in these populations. It's quite a remarkable technique, actually. You can do this based on just, you only need one genome, 2 chromosomes from each individual. In this case, we did it for all the individuals that we have. So in this plot here, time is we're going back in time further to the right, and the y-axis is effective population size in units of 1,000. So if you go very far back in time, you can see here, sort of 1 million years ago, we're inferring an effective population size of around sort of 20 to 30,000. Now, effective population size is always much less than the true census population size. That's true of humans and pretty much every species we measure for various reasons. But 20 to 30,000 is a reasonably healthy number, actually. So humans are around about this value or slightly less. But then it starts to decline. It's a log scale on the x-axis. So here's 100,000 years ago. And at this point, you notice, this is when mountain gorillas suddenly take a precipitous decline. And they've existed, surprisingly, for tens of thousands of years at this really low effective population size of only a few hundred or 1,000 or so. And that is quite surprising, because you would think that this legacy that we see of Inbreeding is a very recent thing due to the fact that humans have essentially demolished their habitat in the last few decades. But they've really been living in this way or something like this for much, much longer. So it's true that they have to some extent adapted to that kind of lifestyle. And there may be things that we see, like their propensity to travel long regions for males to leave groups and to move to other regions, and maybe every now and then to exchange gene flow, genetic material with other gorilla species. That could be part of an adaptation to living in very small numbers. It's certainly not clear that inbreeding, lack of genetic diversity, is itself an indicator of likelihood of extinction. In fact, it's not. You know, the correlation is not that straightforward. The much more important thing is just actual small numbers. I mean, low numbers mean that you only need to lose a few people and then you're down and you're gone. But low genetic diversity, not necessarily such a problem, although I wouldn't necessarily say that it's a good thing. So I think I will conclude there with our picture of a gorilla in its habitat. Thank you.